in honor of Rosa Beddington, who was an eminent mouse developmental biologist who made very important contributions to our understanding of how the early lineages of the pre and post-implantation mouse embryo forms. Importantly as well, she kind of made a major contribution to our understanding of how the anterior-posterior axis is set up. Her and her lab identified that the anterior visceral endodome is an important signaling center that really imparts anterior identity and patterns the anterior-posterior axis. So the winner of today's medal is Rasa El-Mentaite, who is a very worthy winner of this medal. She did her MSc in BSc and MSc at Glasgow University, and then I think she moved to the Sanger Center, where she did her PhD with Sarah Teichmann. Rasa has done some amazing work that you're going to be able to hear about, in which she's been using single-cell analysis as well as spatial transcriptomics to really dissect the development of the human gut, and it's really kind of come to shed light to the important heterogeneity that exists within this tissue and how it arises. And importantly, she's also kind of highlighted the clinical implications of this work by, for example, showing how developmental programs that say are reactivated during bowel disease. And hopefully she's going to be telling us a little bit about this in her work today. And we have a beautiful medal. Unfortunately, you can't really see it very well. And I guess the people in Paris and Barcelona can't see it at all, but it was designed with some pictures of Rosa Beddington in mind. And so without further ado, I will invite Rasa to come up and tell us about her work during her PhD. I would also, just before, I would also like to make a big call out to all the other nominees of the award. So Rasa is a very worthy winner, but the other applicants had some very impressive work, and it was incredibly hard for us on the committee to pick a winner. And so I can please encourage everyone who is finishing their PhD in 2023 to submit an application for the Beddington winner that will be awarded next year. So yes, so Rasa, many congratulations. Thank you.
Wow. Thank you so much for the committee and the organizers. I'm so pleased to be here today and to receive this medal. It's surreal. So today I'll be talking about my PhD work. Probably a little bit of a switch of gears, but I hope you will enjoy it. So my PhD topic was mapping the cells of the human gut across space and time. That means a little bit different things in my PhD, but I'll walk you through it. So my PhD project was part of the International Human Cell Atlas project, which is co-founded by my supervisor, Sarah Teichmann. And the main mission of Human Cell Atlas is to create a comprehensive map of all the cells found in the human body. And of course, there's 37 trillion cells in the human body, so this is not a trivial task to create this type of reference. And so, you know, we can sort of break it down into smaller parts. So, for example, we can think of cells as into two functional classes. For example, the primary cells that perhaps perform very specialized, organ-specialized functions, such as neurons in the brain or epithelial cells in the gut that exchange the nutrients. And then another class of supportive cells that really enable the function of primary cells. So these would be fibroblasts, immune cells, and epithelial cells. And of course, there's huge, enormous diversity of different cell types across organs and different states. And so the goal is to really map it comprehensively. Two technologies that we use in the lab and in Human Cell Atlas Consortium is single-cell genomics and spatial transcriptomics. Single-cell genomics enables us to take a sample from an organ or tissue, dissociate it into single cells, and then use computational methods to project it onto a two-dimensional space. Of course, I've seen a lot of nice posters using single cell, and you can discriminate similarities and differences between different cell types and states in this type of map. We complement this with spatial transcriptomics, where we take a sample from an organ and we put it on a barcoded slide. And this allows us to take not only the full transcriptome of a cell, but also the spatial coordinates within tissue. And by combining both of these technologies, we can start at least to try answer some of the questions about what governs cell identity, how do these cells interact in tissues, and how do they influence each other and organize tissues. So why is it important to have a human cell atlas as a reference? Well, of course, it gives us a lot of information of what are the different cell types that are present in tissue, what are the expression programs, how do these cells differentiate, molecular mechanisms that distinguish these cells from the others, their interactions, physical location. But of course, once we have a reference that encompasses diversity of different protocols, cell states, and diversity of sampling cohorts, then we can start querying perturbation data onto this atlas and ask what are the off-reference cell types that we find in disease or perhaps in a different type of perturbation setting. So actually, the way my PhD project came about was that early in my PhD, I was analyzing iPSC-derived organoids from the gut, and I really struggled to interpret that data because that was one of the first times we could see organoid cells through the lens of single-cell technology and this high-dimensional kind of representation of what a cell looks like transcriptionally. And I thought, if I ever want to interpret any of the organoid data, we actually need to first look what the cells look like in tissues in a very comprehensive way. So the idea was to create a gut cell atlas and identify what cell types and signaling circuits drive the emergence and development of these highly complex intestinal tissues, of course, in humans. There are a couple of things that we want to take into account so that this reference would be reusable for the future generations. So firstly, we know that the gut cells change throughout life massively. Epithelium starts as a flat sort of layer of cells. It then folds into crypt and villi, so patterns into these valleys and hills. And of course, we have immune cells that start to organize themselves early in development and then become functional later on in order to, protect us from invading pathogens, but also to tolerate the microbiome that we have in the gut. So with that in mind, you can't really see this, but with that in mind, we took 4 different time points of life in humans. We sampled intestinal tissues from the 1st and 2nd trimester of development, and we also were able to retrieve samples from mucosal biopsies in children 5 to 12 years old, and also from organ donors in adulthood. The second consideration was really this motivation that a lot of rare and complex diseases in the gut manifest in different regions of the intestinal tract. So we wanted to capture this diversity along the whole intestinal tract, and we sampled 3 different regions of the small intestines, colon, as well as the local hubs of immune system, the mesenteric lymph nodes. And for the adult sampling, we're able to retrieve up to 12 different regions of the adult gut. which was an incredible effort from the Cambridge transplant donor team. And so at the end, we got this UMAP of a lot of different cells from development, adulthood, and from children, both healthy and from inflammatory bowel disease, and ended up with almost 500,000 cells from the gut. Of course, around this time, actually, pandemic hit. So I was in a lockdown and I had all the time in the world to analyze the data from home. So this was very convenient for me. And of course, I could probe the atlas by asking a lot of different questions. There's just so much data at your fingertips, and you can ask anything you really want. In terms of developmental questions, these are some of the questions that I focused on. Firstly, really focusing on this idea that I wanted to see what are the progenitors to the stem cells, the adult intestinal stem cells in the gut that express LGR5. And so, of course, that led me to a lot of interesting literature about how the crypt and villi actually forms during development. And we know that from model systems, there's no single way to make a villus or a crypt. For example, in chickens, you have smooth muscle that contracts and really folds the cells into these structures. In mouse, you have these mesenchymal populations that cluster and creates this patterning of restricting the proliferation of some of the epithelial cells and allowing this kind of pattern to exist. And so what I was able to do with our data is to define the transcriptional profile of these cells that are before they transition into crypt and villi, and track some of the genes that are expressed at that stage and how they are expressed at a stage where we already have crypts present. So for example, you can see BEX5 that maps, which we probably can't see very well, but the BEX5 gene that mapped these uniform progenitors at early embryonic stage were also still expressed at the LGR5 stage, which kind of that and a lot of different single cell analysis suggested that actually these transition into these crypt epithelial cells. I've also looked, of course, because the cell signaling is so important early in development, I used these algorithms such as cell phone DB developed in Sarah's group, where we can ask the question, if the ligand is expressed on cell A and the receptor complexes are expressed on cell B, we're more confident that these are the cell types that interact in space. And by parsing through a large data set of known ligand receptor interactions, we're able to point which are the interactions that appear at these early stages. So of course, something like BMP and PDGF signaling, of course, appeared. This is well studied, but we also had some other signaling that we were able to point out in our analysis. So sort of a quick overview of this part, and this was the main conclusion was that the epithelial and fibrillous smooth muscle cell interactions both support crypt villus formation in humans through the lens of single cell data, of course. Then the second topic was really looking further into how the enteric nervous system develops. That's a completely different cell type. These are neuronal cells, the second brain that exists in the gut, our second brain. And we know that these cells colonize the gut early and during development. And these are the images of small and large intestines of small and large bowel from human embryos, where you can see these enteric progenitors starting to colonize the mesenteric plexus at 6.5 post-conception weeks, earlier in the small intestine than they do in the colon. And so for this part, I used an algorithm, SC velocity, which takes into account the splicing and the ratio of unspliced and spliced mRNA reads in single cell data in order to infer a pseudotime trajectory from this data. And looking at the differentiation of these enteric neurons, I was able to point out that there's two different stages of these enteric progenitor differentiation in humans. Firstly, going into a neuroblast and two branches of neurons. One is inhibitory neurons and the other more motor neurons based on their transcriptional profiles. And then later on, this differentiation becomes more balanced and you give rise to also glial progenitors. So this is illustrated in the graph. And the third part of my PhD thesis was looking at the gut-associated lymphoid tissue formation. This project started because when I was analyzing Crohn's disease data, we found a small population of mesenchymal cells that seem to look like these lymphoid tissue organizer cells. And so it seemed like a perfect opportunity to look into this developmental process of how lymph nodes and gut-associated lymphoid tissues form during development. Here you can see an image of 8 post-conception week human gut. At this point, we don't really see any lymph nodes forming, but at around 16 post-conception weeks, we could already see these hubs of cells in the mesenteric endothelial beds. And we were able to dissect them and profile the cells in these hubs separately. So I relied heavily on what is known in the literature about how the lymph nodes form during mouse development. So we know that there are 3 different cell types that are important for organizing a lymph nodes in mice. There's lymphoid tissue initiator cells. These are ILC3 cells. We have 2 different types of lymphoid tissue organizer cells. One of them are endothelial in origin, and the other ones are mesenchymal in origin. And through the stepwise interaction with these cell types, the mesenchymal lymphoid tissue organizer cells become activated, and they start expressing chemokines to recruit T cells and B cells into these local hubs to create lymph nodes. So I began by looking for the last step within this process, so the mesenchymal lymphoid tissue organizer cells. And there was a small population of these cells highlighted by activation signature that was present both in mesenteric lymph nodes from developing fetus and also small and large intestines. I could also see that there's a similar population transcriptionally that is endothelial in origin, and it shared the adhesion and recruitment signals with the MLTO cells. So we sort of postulated that perhaps this is another cell type involved. And then when I looked for ILC3 LTI-like cells, this was actually quite difficult to understand because there were 3 different states that I could see in the data. All of them are ILC3-like. They have characteristics that have been described for LTI-like cells. But they also had separate characteristics, and we were able to image them in the fetal gut almost next to each other, expressing the key marker genes that are differentially expressed between these states. Of course, the next step was to understand whether they actually are spatially located in the same place. And we use spatial transcriptomics here, and we're actually quite fortunate to capture a region where the MLTO cells sit. So this is the region that is where there's expression of CCL21, CCL19, and CXCL13 characteristic of MLTO cells. And of course, by using combination of single cell data with the spatial transcriptomics data, we could deconvolute precise cell type states that are present in this region. And what we could see is, in fact, the population that we postulated was present in that region, as well as LTI-like NCR positive ILC3s. But interestingly, nearby, there were also the microfold cells, which are suggestive of these structures in the adult intestinal tract. We could also see these cells starting to home in using SM fish RNA-scope type of technologies. So we could see single cell resolution where these cells appear in the intestinal tract. And since we have this observation of, similar populations emerging in the inflammatory bowel diseases, we asked the question of whether these mechanisms could be relevant in our understanding of inflammatory bowel disease. And for this, we used IBD GWAS associated hits and looked for the enrichment of the set of genes in our data in both genes associated with Crohn's disease and with ulcerative colitis. And we could see that there are a lot of different cell types that have enrichment of genes, but of course, part of genes were also enriched in these ILC states that we see during development. And long story short, we sort of hypothesized that the cells that are involved during development and recruitment of these immune populations during development are also important for retaining and recruiting immune populations into the inflamed gut. So there's characteristics that are shared. Overall, this is sort of the model that came out of the study and my PhD, where we have 3 key cell types with different substates of LTI, LTO cell types that together initiate the recruitment and retention of immune cells in the gut during development, and then also similar states exist in inflammatory bowel disease. Instead of a summary, I just wanted to finish up by saying that you can continue asking questions because all of this data is available. And you can go and browse at Katsalatlas.org. And thank everyone involved. So of course, Sarah Teichmann, who's my supervisor, a lot of different members from the lab. Of course, it takes a village to make this type of data. So I was very fortunate to be part of this project. Thank you for listening.
So thank you again, Dasha, for the part of the talk. Congratulations.
Thank you.
Do you have any questions? So okay, so in the meantime, while people think of their questions, I will start by asking. So very nicely. Hey, I was going to ask you to repeat the question. Yeah, so you very nicely show that in the inflammatory bowel disease, there's this recruitment of and retention of these lymphoid cells. So do you have any idea, because obviously the retention is probably aberrant retention, that leads to the prolonged phenotype of inflammation. Do you have any idea of what causes this prolonged retention that could potentially lead to the disease phenotype?
So in fact, this is something that we're working on right now. So we know that actually a lot of therapies that prevent immune cell recruitment or initiate retention in the lymph nodes help the IBD patients, and so we're sort of initiating more studies and in profiling the inflammatory bowel disease patients, and also longitudinal profiling of these patients, because that would allow us to sort of time point when something gets dysregulated and how that compares to developmental programs. But of course, in our study, we're able to only get the IBD samples from the children. So it's also a limited amount of what you can do. But yeah, to be asked further. Thank you. There's no questions.
I'll ask another one. So in your developmental studies,
Yeah.
you I think you very nicely showed how, let's say, the emergence of these lymphoid cells could take place. But have you, I think I may have missed it, have you applied the cell chat algorithms to how these cells communicate with those what interesting insight did you gain from this analysis?
We did cell phone DB on these data sets, and of course there was a lot of signaling that has been partly has been described in mice from some of the interactions that I showed in the beginning, the LT receptors interacting with these cells interacting through those receptors. And of course, there's many other. The thing is with single cell data is that you have so many, and it's narrowing it down to the really interactions that you care about, and you can go on and further validate and probe in model systems. And I think it's also key. So there were many. To be honest, it's been like a year and a half, so I can't remember exactly the receptors, but happy to chat more about this.
Okay, then. So I think we will call this session to the end, not before congratulating Rasa once again for her. And I think Paul wants to say something.
Yeah, so PhD prize talks are fantastic, aren't they? That was a cracker. That was the British Society for Developmental Biology's PhD prize talk. Tomorrow morning at 9:30, the French Society for Developmental Biology are going to award their equivalent PhD prize, and you must come to that talk. It will be streamed in at 9:30. In the sort of smell of Europeanness, at 12:30 we have ERC coming for an hour. Sally knows more about this than me. Come back here, but can they come back with their brown paper bags? Are we allowed to eat, do you think? 10 minutes and then come back. Now, it's very timely. You all know that the UK is back in business with ERC sort of stuff. But everyone across Europe needs to know how to get these grants. And then this afternoon, and Sally is going to be our chair. We're streaming in not from France this time, but from Barcelona. And their chair is who? Well, they will have a Spanish person chairing, and Sally's going, and we're going to do exactly as we did with the pasta yesterday. We're going to interdigitate a talk from Spain, a talk from Oxford. Fabs. So go and get your brown lunch, eat it quick as you can, come back here for some ERC action. Shall we say, oh God, you decide, Shankar. 12:35 back here if you want to know how to get millions of euros.